Hello everyone, Dr. Reza Rezarei here from Cyagen. Today, we're diving into the world of retinitis pigmentosa, a constellation of rare and inherited disorders that casts a shadow over the vision of those it touches. RP marks its presence by a gradual but relentless decline in sight, often culminating in complete blindness. The journey of RP begins with night blindness and narrows the field of vision over time, akin to looking through a tunnel, until little to no vision remains. Central to the story of RP is the RHO gene mutation, a key player in this condition. This mutation affects the rhodopsin protein, essential for the photoreceptors in our eyes to capture light and initiate vision. When this gene malfunctions, it triggers the progressive loss of these photoreceptors, starting with the rod cells crucial for seeing in low light, and eventually affecting the cone cells responsible for our ability to perceive color and fine detail. In our pursuit to combat RP, Cyagen introduces the HUGO-GT model, specifically the B6J-hRHO mice. This model is engineered to closely replicate human RHO gene mutations associated with RP, serving as a critical tool in the development of gene therapies. Unlike typical models, the B6J-hRHO mice express the human RHO gene, maintaining normal retinal structure and function. This unique feature makes them invaluable for studying RP and testing new treatments. The B6J-hRHO mice stand out for their robust immune response, comparable to wild-type mice, but with a critical advantage: they express humanized rhodopsin, offering a direct pathway to exploring RP's genetic basis and potential therapies. Our model undergoes thorough validation to ensure its effectiveness and relevance in retinitis pigmentosa research. Detailed analyses reveal that the B6J-hRHO mice exhibit normal retinal morphology, mirroring the ocular structure found in wild-type mice. This includes assessments of the fundus and retinal vasculature through optical coherence tomography and fluorescein angiography, confirming the absence of significant changes in ocular and retinal morphology and the maintenance of normal retinal vascularity and blood circulation. Furthermore, the expression of rhodopsin protein, a crucial element in vision, remains unaltered when compared to wild-type counterparts. This observation comes from meticulous hematoxylin and eosin staining and immunofluorescence staining analyses, underscoring the intact retinal structure and functional expression of rhodopsin in these mice. Electroretinogram testing further substantiates the functional integrity of photoreceptors in the B6J-hRHO mice. Both scotopic and photopic ERG recordings exhibit A-wave and B-wave amplitudes nearly identical to those of wild-type mice, indicating that the retinal photoreceptor function in this model is preserved and operates normally. These findings, derived from a combination of advanced imaging techniques and electroretinogram testing, affirm the B6J-hRHO mouse model's validity as a powerful tool for studying retinitis pigmentosa. It replicates human RHO gene mutations' effects on retinal morphology and function, thereby offering an invaluable resource for the development and evaluation of gene therapies targeting RHO mutation-associated RP. As we delve deeper into our fight against RP, it's crucial to acknowledge the innovative approach behind our HUGO-GT program. Humanized mice have emerged as the preferred tool for disease research due to their strong correlation in mimicking human physiological and pathological characteristics. Yet, existing partially humanized mouse models have limitations in exploring genetic diseases' pathogenic mechanisms or drug development, such as random insertion complexity and insufficient humanization areas. To comprehensively study these mechanisms, we need mice humanized with long fragments or even the whole genome. Cyagen has initiated the HUGO-GT program, employing our proprietary Turbo Knockout Pro technology for in situ replacement of murine genes with human counterparts, successfully creating whole genome humanized mice covering a broader range of intervention targets. The HUGO-GT mice incorporate an efficient large fragment vector fusion technology, serving as a universal template for custom mutations, providing a preclinical model closer to real-world biological mechanisms for drug development. Furthermore, the HUGO-GT program extends beyond offering models. It enables comprehensive CRO services in fields like ophthalmology, neuroscience, and oncology immune diseases, empowering genetic disease research and gene therapy drug development. With the HUGO-GT whole genome humanized models, we're not just understanding RP but actively seeking transformative solutions. Thank you for joining me in this journey into the heart of genetic research. The B6J-hRHO mice, part of our broader HUGO-GT initiative, are more than just a model. They're a beacon of hope in our quest to understand and ultimately conquer retinitis pigmentosa. Keep following Cyagen for the latest insights and developments in our HUGO-GT models as we continue advancing the frontiers of genetic research.